Let me first introduce myself. I am Jana, third year PhD student at the Clinical Child and Family Studies Department. And under the supervision of Marianne Bakemans-Kranenburg, she's also in this session, and Mirjam Oosterman. And today I'm going to present to you the pre-registration of my next study on the effect of the video feedback intervention on children's hair cortisol. So I'm gonna not look at the parenting effects, but I'm gonna take a step further and look at child outcomes. Yeah, and you have just heard kind of everything about the intervention from Kim. So I'm gonna be very, very short on that part. So just keep in mind that what Kim told you about the intervention is basically all true, also true for the intervention, but then in a parent-child context at home. Okay. So why cortisol, you may ask, and I think in this field, not a lot of people are really focusing on cortisol. So I'm going to give you a quick overview on what cortisol is and does, because it's quite an important hormone in the body, and it has many general functions, but it also plays a role in the response of the body to stress. And if you're in a stress situation, for example, you encounter this very beautiful female lion and she wants to eat you, then your hypothalamus pituitary adrenal axis, the HPA axis, becomes active. And it starts here in the paraventricular nucleus, and that releases the CRH and the pituitary gland, which is also in the brain, then activates the production of ACTH. And the ACTH travels to the adrenal gland, and that triggers the production of cortisol. And usually there's also like a negative feedback loop showing the brain, okay, here's enough cortisol, you don't have to take the next steps to get more cortisol produced. But well, this is really helpful for the body because you have to run very, very fast to not be eaten by this lion. But if the HPA axis is deregulated, for example, by continued or frequently repeated stress, then basal cortisol may be chronically secreted in excess. And that has been related to many negative outcomes. For example, yeah, well, this is a really, really long list of different outcomes. Due to prolonged cortisol exposure. And we find many physical health effects, for example, cardiovascular disease or diabetes, but we also find mental health aspects and behavioral or emotional aspects in children. For example, externalizing behavior, and later on in adults also depression and anxiety. So this is really not good. We don't want that. But, well, always look on the bright side, because if all the negative environments or all the stress can have a negative effect on cortisol levels in children, maybe a positive environment can also have a positive effect. So besides those extreme influencing behaviors or factors such as war or child abuse, parenting might also have an effect on children's cortisol. And instead of focusing on the negative impacts, parenting interventions aimed at improving parenting might also have a protective effect on cortisol levels in the children. And there is indeed some findings in the literature. For example, the ABC intervention was conducted, and in 4 to 6 year old children who were involved with child protective services, they found a more typical diurnal salivary cortisol levels when these children were in the intervention group compared to the control group. And also Marianne and colleagues found in 1 to 3 year old children with high externalizing behavior and the DRD4/7 allele that diurnal cortisol production decreased after the FIB intervention. By the way, I call it FIB. I'm not sure what's the correct one, but I think it might be difficult for me to adapt right now. And last but not least, one randomized controlled trial on 39 parents and their children. They were between 4 months and 5 years old. they found that children's hair cortisol concentrations decreased significantly in the intervention group compared to children in the control group. But there's also a meta-analysis on 19 intervention studies, and they did not find a significant overall effect of parenting interventions on children's cortisol levels. So that's kind of like, okay, what's going on here? One possible explanations for this might be the differential susceptibility hypothesis. And although parents, of course, play a crucial role in shaping the experiences and the life of children, children might vary in how much they are affected by positive as well as negative parenting. Think of dandelions and orchids. And dandelions, they grow basically everywhere, and they do not really care about the ground they're in. Whereas orchids, they need a very specific environment to flourish. But when they do flourish, they are also exceptionally beautiful. And according to this theory, highly susceptible children, so orchids, they shall, when they are exposed to negative parenting or negative environments, show more problems, but they also shall flourish more than when they are exposed to supportive environments. Whereas the dandelion children, they will basically stay the same throughout different conditions. Well, and we want to investigate this. So I'm part of the big LSIT study, and it's a large longitudinal twin study with 2 different cohorts. And we followed the children over the course of 6 years with alternating home and lab visits. And for this study, we had 1 pre-measurement. And then we conducted the VIP intervention, which was also a randomized controlled trial. And 1 month after the intervention was conducted, we had the post-measurement. We did that in both cohorts, the early childhood cohort and the middle childhood cohort. And to give you an overview of both samples, the early childhood cohort consisted of around 200 twin pairs, so 400 children and their primary parent. They were around 4 years old at the beginning of the study, so about 2 years older when they received the intervention. Almost half of them were boys, and a bit more, half of them were monozygotic twins. The parents, well, the primary parent, so the parent that spends most time with the child or the children, was predominantly the mother. 37 years old on average, and this sample was quite high in SES. So most of them were medium to high SES. And we see a similar picture in the middle childhood cohort. We had some more children, so 243. They were older, of course, because it's middle childhood. They were 8 years old on average at the beginning of the study, but sex and zygosity were almost the same in both studies or in both cohorts. And also,
Jana, just to say that you have 5 minutes left.
Oh yeah, that. Ooh, okay, I'm gonna be very quick.
Speed up a little bit.
Yeah. Well, the same for the parents. Yeah, and I'm not going to talk about that because you already heard it. We conducted the VIP intervention study. Well, how did we measure cortisol? Many previous studies always used saliva, but the thing is that salivary cortisol provides kind of a snapshot of cortisol levels at this particular time point. And cortisol levels vary highly from day to day. They are affected by food intake, by activity, by lots of different factors. And well, I have been talking about chronic stress. So high cortisol at one time point is not really indicative of chronic stress. So a complementary approach is to use hair samples because hair cortisol is quite stable over time, and it's a really good way to assess retrospective cortisol levels. So that's what we did in this study. And the differential susceptibility study measure, we want to use a polygenic score of differential susceptibility. And this is, I think, for many of us, quite something new. We can get those scores by using genome-wide association studies, because all humans have like near identical DNA sequences, but there are slight differences between individuals, so making each of us very unique. And these differences, they're called genomic variants, and they occur at specific locations within the DNA. And in genome-wide association studies, researchers identify those genomic variances that are associated with differential susceptibility in our case by comparing the genomes of individuals with or without this susceptibility. And on the basis of those GWASs, we can estimate kind of a risk score in how much susceptible you are to the environment. So this is then basically the polygenic score, which we will be using in the analyses. Yeah, and the model we will be running is quite simple. We have the intervention, though they're either in the intervention group or in the control group, and we will see whether their hair cortisol after the intervention will be significantly different between the groups. And we will use the polygenic score of differential susceptibility as a moderator. And to add a bit spice, well, we will use intent to treat analyses so that we include all of the participants that were randomized into either the intervention or the control group. And we will use multi-level modeling because, well, as I said in the beginning, we have twins, and those are, well, nested in one family. And of course, cortisol is also highly genetic. And they would be very much similar to each other. So we have to account for that. And with that, I'm at my last slide because I haven't done the analysis yet. We are still waiting on the genetic analyses of the DNA samples. But I hope to be able to analyze and write the things up before the summer. So we will hear from me. And well, that's it. And I'm happy to hear some questions.